Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Before we jump into this episode, I'd like to share a few things with you. The December and last Dr. GPCR newsletter of 2020 is now available. If you haven't subscribed yet, please visit drgpcr.com/newsletter. Also, don't forget to share the newsletter with your colleagues. We cannot wait to bring you brand new Dr. GPCR newsletter editions in 2021. Second, we will be taking a short break of the Dr. GPCR podcast in January 2021 as our family will be welcoming a baby boy. But rest assured, we are already working on bringing you brand new episodes, starting sometimes in February 2021 with an amazing lineup of guests. Also, stay tuned for a major announcement regarding the podcast. We're also proud to announce that we are pursuing consulting opportunities in the GPCR field. For help with your R&D project, please visit drgpcr.com/consulting or reach out by email at hello@drgpcr.com. And last but not least, we will be closing this first season of the Dr. GPCR podcast with a series of interviews with phenomenal female scientists in the field. We hope you'll enjoy it. Hello, everybody. I'm Dr. Yamina Bershish, founder of Dr. GPCR. Welcome to another episode of the Dr. GPCR podcast. Today, I have the great pleasure of talking to Dr. Maria Waldhoer. She is currently the CSO of a company in Switzerland called Interax Bio AG. Welcome, Maria.
Thank you. Thank you for the invitation. Hello, everyone.
Thank you for coming on. I'm so happy to talk to you today.
It's wonderful to be invited. It's actually a great honor to be invited by Dr. GPCR to talk about GPCRs.
Thank you so much. Thank you so much. I'm excited to chat with you today. So as I mentioned, you're currently the CSO of Interax Biotech AG. Can you tell us a little bit more about your career and how you got to where you are today?
Yeah, thank you for asking. It's actually been a long way through the bright and the dark side of science. So I actually started studying biology, zoology. Not many people know that. I'm not a pharmacologist by training. I'm a zoologist by training. Back in Salzburg in Austria, where I'm from. And I studied neurobiology as a special topic there. After my master's thesis, I decided I actually want to do something different. And I started in the lab in Vienna. But first, before I actually started working on GPCRs, I started working on cell cycle regulation. Because the professor, Misha Freismuth, who I worked with for many years on my PhD, then he had a postdoc at that time, Veronika Sechsel, and who started working on this. And I started with her. She left for a postdoc, and one day Misha came down to the lab and said, "Maria, now you work for me, and you work on GPCRs." And that's how the whole story started. It wasn't even me picking this. And then I did my PhD in Vienna with Misha and moved on, did a postdoc in Copenhagen in Denmark with Ture Schwarz, who many people know. The Great Dane, I call him, and had a lot of fun there, had a really great time there, and then decided to do a second postdoc with my very good friend, Jennifer Whistler, who is now at UC Davis. Mm-hmm. And had another grand years there. And at that point, I was ready to start my own lab. And I went back from San Francisco, where I was with Jennifer for about 4 years, and started my own lab in Graz, which is a little bit east of Vienna, actually southeast of Vienna. And had my own lab and thought, that's it. I'm in academia heaven. You know, I went all the way up to be an associate professor. And then midlife crisis, I turned 40. I thought, that can't be all. And the dark side called. And that was when I started at Novo Nordisk in Denmark. Mm-hmm. R&D in early drug discovery, working on incretin receptors. And that was the ride of my life. It was so much fun to actually start applying, translational ideas and start really working on drug discovery. And I was there for 6.5 years. But all good things pass, and I got antsy again and got headhunted by Gebhard Schertler, Professor Gebhard Schertler, who at that time was in Switzerland. And he called me one day and he said, "Maria, I have a small biotech startup and they need someone to show them how to pipette." So Not really like that, but he said, Maria, I think these are three founders, and they are just out of their postdoc, and really would need someone with a bit of industry experience. And this is how I came to start at Interacts Biotech in 2017, and have been there ever since, and started developing a slightly new technology, which I will talk about it a bit later.
Yeah.
Together with especially Aurélien Risk, who is the CTO of the company. And as of now, we are there for 3 years. And it's up and running. We are 15 people. We started being 4. So it's good fun. So that's me in a nutshell.
That's awesome. That's awesome. I really enjoy listening to these stories, these trajectories. A lot of times people have this story about, going to a class or listening to a talk and saying, wow, I heard about GPCRs, but I think you're the exception so far.
No, not at all. I was like, who cares about GPCRs? I want to work on cell cycle regulation, right? And but I've been doing this for more than 20 years now, and I still love GPCR. So there you go. It was a real fate.
Yes, and thank God you were told you have to work on GPCRs now, because you kind of adopted them, and this is what you got. Throughout your career, so you have the industry experience, you also have the academic experience.
Yes.
When you moved from academia to industry, were there any challenges that you faced? You mentioned that you really loved working in industry and working on the translational aspects.
Yeah.
Can you tell us a little bit more about that transition?
I think there are pros and cons. I think I actually was an untypical academic. I like speed, right? I'm not the most patient person. People who know me will laugh now because I'm really impatient. So I want quick data, good data, high throughput, and the speed of how projects were run in a big company, that was exciting for me. There were so many different people working, so many really, really cool different people working on one big project, and that really enticed me, I have to say. Challenges, so that was the beauty of it, and decisions were made. Now you go for it, you get this kind of personnel, go do it. There's the flip side, of course, when management decides, as wonderful as it sounds, it doesn't fit in the overall agenda of the company, we have to stop it. I was in tears. It happens to everyone. It happened to me as well. It's a big clunk on your head. It's like, now you have to stop doing what you had so much fun doing, do something different. And I think that's the most difficult part in big pharma, I have to say, that you have to do in the end what management tells you to do. This is why I left and started doing management myself.
Yes, this way you
biotech, because now I can decide what's going to be done, more or less.
But so, you have a bigger say in where to.
Absolutely, yes. But biotech, on the other hand, you'll have to be even faster, and I think what really prepared me for it was that both in academia you learn how to write grants, you know how to do science really well, you do all these kind of things. In big pharma, I didn't have to write a grant. I had to apply for some internal funding, but usually, it was some sort of manager that helped you do this. You just had to convince upper management that this is a good idea in the boardroom once in a while. That was about it. In biotech, you have to do both, right? You have to set up the entire science and control it, and you have to go for funding, and you have to go for big funding.
Yeah.
So I think it really prepares you, the career that I had, it really prepared me to actually now be fast and quick, and you have to convince not only the scientists on the scientific grants, you also have to convince stakeholders and investors. Yeah. You have to talk to people that have no idea what you're doing, but have a lot of money, and you need to I wouldn't say dumb down, but sell your product in a way that your grandma would understand it. Yeah. And that's a huge challenge, I have to say, but it's also really good fun.
Yeah, and it must be, especially with the experience that you had. So you had the experience in academia, where usually when you apply for grants, you have to be as complex as possible and innovative. But then again, getting the big pharma experience, then getting the biotech experience is also a plus.
Because I think that was a huge plus. Yes. Yeah.
That sounds like fun. So tell us a little bit more about Interacts Biotech AG.
Okay, AG is just like the AS in Denmark or whatever. It's
Okay.
Index Biotech. So it actually started out. There were three founders, and it started out on something completely different. It started out on using arrestins as biosensors. And Martin Ostermeier, he was one of the founders. It was his PhD thesis. He actually started to accrue money for this and interest in it, and he started very successfully this company with two others. It soon turned out that that was really tricky to do and very hard to do, and there were a lot of competition out there. Just another biosensor wouldn't make a company run. What I have done already at Novo Nordisk was I became interested in analyzing in vitro data in a slightly different way. We all know bias it was, I would say, the big en vogue thing at that time. But we at Novo Nordisk, already a small group, we already started analyzing not endpoint assay data, but kinetic data of ligands and how they activate pathways. And we hired a super postdoc, Susanne Roth, who now works with me at Index Biotech, to actually start modeling signaling pathways on the computer. Wow. Using and everyone who I talked to in the beginning said, "You're completely mad. You know, this is ridiculous." Stephen Hill is a very famous one who said, "Oh, you are mad," but now he's a fan. So what we do is we actually start a little bit like when you build an airplane. You never build an airplane from scratch and then look if it flies. You first model it on the computer, okay, and you model how the aerodynamics should flow. This is a little bit of how we do it with the target, with the GPCR target and the signaling pathway. We model the pathways on the computer with mathematical equations and look at the interactions of them thereof in time and space. And then we build a wind tunnel, okay, to test this. And the wind tunnel in our hands is actually cellular assays, very well controlled kinetic signaling assays. And then we refine the model with data that we get in our wind channel in our cellular assays. And in the end, we can describe compounds in much more detail than just getting efficacy or potency or even a bias factor, because we look at how these pathways interact with each other over time. So we can give you rates of, cyclic AMP, internalization rates, recycling degradation rates. We can give you the affinity of G proteins for a receptor ligand complex and stuff like that. And we can actually call it fingerprints. Okay, we get pharmacological fingerprints. You, instead of 3 parameters, you get about 10, 15 parameters for really one compound on a given receptor. And this is actually what we do. And we were very lucky to actually do a couple of really cool projects with big industry. Boehringer Ingelheim is one of them. Another one is Lundbeck in Denmark.
Wow.
And right now we are working with a South Korean company called GPCR Therapeutics. And what they use us for is they give us late stage compounds that they want to test in vivo, and they want to get more information on them. And this is what we can do because we can describe this very well with our computational and also cellular assays. So this is our platform that we started out doing. Mm-hmm. And then, of course, and I think it was one thing on the plate. What is new and exciting in the field? AI is a big en vogue thing.
Yes.
Right. However, AI, I like to say we combine AI with RI, with real intelligence. So what we want to do is we want to take the information we get out from the descriptional parameters from our systems biology analysis, and then use AI algorithms to then design compounds that do pretty much the same thing. So we want to be able to predict residence times of ligands. We want to be able to predict certain properties on a GPCR of these ligands and use AI. And we actually have first successes doing that. So this is what we are doing right now to implement that because we were asked by customers. So now you describe our compounds for us. Can you assist us in making them better? And that's what we are doing and focusing right now on.
That is extremely exciting. I think it's something, measuring the kinetic response of a ligand and how the activation of multiple pathways happen when you treat your system with a ligand has always been a very difficult thing to look. Not only because you couldn't analyze the enormous amount of data that you got out of it, but then you didn't know what to do with it.
Exactly. Yeah. And bias, the whole bias factor, to be honest, bias was there when I was a PhD student in Vienna. It was called functional selectivity.
Yeah.
Okay. When I started working with Jennifer, she had Rave, which is receptor activations versus endocytosis. Then it's always been there, right?
Yeah.
So, and then people, I think, made a little bit, you look at arrestin activation after whatever 10 minutes, you look at cyclic AMP after half an hour, depending on what your assay kit says. For some of them you have to overexpress GRKs. You completely mess up the system. You cannot compare apples and pears. You can get a hint of what's going on. But you have to also look at the speed of the signaling pathways. Calcium is very fast. Arrestin recruitment can be very fast. Cyclic AMP is ongoing over time as long as your ligand is there. And this is what we try to capture with our systems biology analysis, by you looking at kinetic traces of different pathways, analyzing them at the same time, okay? And look how over time these things interact with each other and change, and then read out the parameters from the computational model. And we get really exciting stuff out. We can predict partiality of ligands, even if you're in an overexpressed redundant system. So we can do all these kind of things, and that's super exciting.
Wow. And then the AI's role in this is not only analyzing this data, but also learning from it. And
using this data pool to actually then generate the fingerprints for the compounds. Yeah.
Wow, that's fantastic. And can you tell us a little bit more about? Well, I think it's evident for our listeners that this is very cool and it helps advance drug discovery. But
yes.
Can you comment a little bit more about how do you see this technology, take us to the next level when it comes to drugging GPCRs?
Well, I think we can speed up the process. I think actually designing drugs with certain properties that right now you say most potent that works. Ligand residence time is an important thing, because it's not only the PK in the body that determines what the drug is going to do. It's actually also what is actually the drug doing at the receptor. So I think many big pharma actually start working on these new technologies, and they all jump on AI, but they jump on the big mass of structural data, okay, and not so much on the functional data. So I think putting the functional data in there, that will be a game changer if we really succeed in it. I'm not saying we are there yet, okay. So I think this can change the picture. And I've been talking to a lot of big pharma representatives recently at the Bio International online, right, thanks to COVID. And let's say when I started this job as CSO, a big pharma would say, oh, no, we just want that compound and in-license it. And now they came and they said, okay, Maria, we are really interested in novel disruptive technologies that do something different that no one else has yet. And just having a normal AI algorithm based on virtual structural data is not good enough anymore. So I think it can be a game changer. Whether it's going to be true, that's the other question. But we are not the only ones doing it. So more and more academic labs start working on it. I know big pharma's labs also start working on it. We have often when we go to customer talks, there are these geeks in there, right? That they say, oh, this is really cool. We're doing similar stuff. Right. So I think this is something to use the computational power is really important now that we have all the structures, right?
Yeah. All the structures, but also all the tools in the lab as well, where you can run X number of plates in a couple of minutes.
Yeah.
Having to pipette these.
But I actually hope that this will be not necessary so much in the future. These high throughput screens or fragment screens or stuff like that. So I can give you an example. We had a project with an academic colleague working on just to show that we can just make some stuff based on virtual screening. And that took about half a year with all the docking and stuff like that. And now with our AI approach, we could do this in 2 months. And we had hits, and we actually had agonists on the receptor, not antagonists like before. So I can tell you this is really speeding up the system. But it wouldn't be possible without the structural information. Okay, as much as people that know me, they know when I see a structure, I just was like, okay, whatever, it's another thing, right? I'm more the biology functional person, but when I was in Novo Nordisk, I got really excited because I worked with very, very cool structural biologists there. And I got really excited when they explained to me, this thing is then tweaking in there, and this is why. So I kind of started getting it. So I think overall, it's where we are today. It gives us so much more opportunity to actually speed up the process and make actually drugs with less side effects, or at least propagate them in that direction before they even hit an animal, to see whether or check out whether they're off-target effects. That's also what we can figure out with our systems biology approach. We can find out when a compound doesn't follow the pathway, which means it has off-target effects. Wow. It could be it's just stuck in the membrane, or it's hitting something else, right? So that we can also do.
But that's the kind of information that you really want to know before you advance that compound, and it ends up in an animal, and definitely before it ends up in a human being.
Yes.
In that sense, yeah. I was, as you know, we have a monthly newsletter. Yes. And last month was a structural month. I think there were 10 papers published on a variety of structures. And I remember when in 2000, the first crystal structure for Dobson came out, and it was this big hit. And right now they just come out almost on a monthly basis, which is fantastic, because then you can use the structural information, you can use computational tools, and at the same time you also need to understand the biology, the underlying biology in a pathological system, and kind of putting these together will definitely help streamline drug discovery and target.
Absolutely, yeah.
What are some of the most interesting new developments in GPCR drug discovery, if you can tell us a little bit about that?
I think I just mentioned that, right? I think it is to actually jump on all the information that we have, and really, go for the more multiplexed, a picture, I have to say, right? I remember one funny thing, dimers was a big thing for a while, and I mentioned dimers because I think now we know so much about receptors, what they do individually, right? Yeah. But if you look, and now I'm also jumping a little bit forward, if you look at what is the big challenges that we face when we work in GPCR drug discovery, or in drug discovery altogether, what are the big things right now that we need to treat? And I think comorbidities and aging population is a huge issue, right? People get older and older. So we need to actually start looking at the interplay of the individual receptors with each other, whether they are dimers or whether there are 15 receptors co-expressed of different kinds or different subtypes in one cell type, or whether the relationship of them to each other changes in pathophysiological or diseased tissues. This is something really, really big. And I think this is where we have to go now with the computational power that we have to also go in looking at these kind of things. Okay, not only at your one receptor, your one drug hitting it, it's how do these all interchange, right?
Yeah, and I think and you're right, that's where the computational power really comes together. And if I can add to that, I think also being able to understand how the landscape changes in a disease state.
Yes,
in
exactly.
A patient who has chronic disease, that's also very important for drug discovery, because you would think, at least logically thinking, someone who's healthy may have a different set of receptors or different cells than someone who has a chronic disease who's already been taking some kind of medication over a period of 10 years.
Let me give you one example. When I worked at Novo Nordisk, the in vivo pharmacologists would always say, you and your HEC cells, okay, your HEK cells, you artificially overexpress, and we just need it for potency, right? So, and then you have to do this with the endogenous level. And then my counter question was always, what endogenous level would you like me to mimic? And then came the big question mark. It's like, I'm like, do you want to have this in a, pancreatic islet? Do you want to have it in a, stage whatever diabetic? Do you want to have it in an obese person? Do you want to have it in the brain area or wherever this receptor is expressed that you're interested in? And how old should it be? And what is your endogenous level you want me to mimic? And it was always funny because there was no answer.
Yeah.
Right?
Yeah. And I think there still is no answer, at least a very hard question to answer. And the Hexel question came up in many of my conversations in the context of the podcast. And yes, they're Hexels. It's a quote unquote artificial system, but it's a simple system, and they grow fast, and it allows you to get information, at least basic information.
I think it's important that you understand the basics of what you have and what you're working with. You need your basic information, and then you can think bigger, right? So I did a really cool project there, and it was probably my most exciting scientific ever thing to find out, based on a theory that we had. We ran a project at Novo Nordisk, and in Hexels, and we designed the compound to work like exactly this in a HEC293 cell. And then we put it in the animals, and it worked. It did exactly what we wanted it to do. And this was probably the biggest moment for me as a scientist, because I worked on this project for a couple of, more than months, I would say, and we put it in a mouse, and then I got the call from the vivo pharmacologist, and she said, Maria, this is amazing. The effect is just like black and white. Wow. And that was a big moment. So I said, with my little tiny hex cell and a bit of theory behind it, we managed to make this compound. So that was really exciting. So it's you can learn a lot from analyzing your compounds in a tiny, lovely hex cell.
Oh, definitely. That reminds me of a collaboration I had. To make a kind of a long story shorter, I met someone at a conference who saw my poster and said, "Well, you have, a cool system. You're using biosensors to measure receptor function. Yeah. Let's chat." And so what we were able to show is that a mutation, a single point mutation in a G protein, ended up uncoupling it from its receptor. Yep. And what it helped us understand is the mechanism on how this mutation that was expressed in only 7 people or 8 people now in the world was affecting their immune system and their immune response.
Yeah.
And that was the first example. I think this is the only example. Now I have yours that shows that, you can use a very simple system to understand something that happens in a human.
Absolutely.
And we were able to kind of describe on a molecular level why it was happening in these patients.
Absolutely, yes. That's why when I did my second postdoc and also at Tua's lab and at Jennifer's lab, I love to swap around C-termini from one receptor to the other. The tail swap experiments. It's so much fun doing that, right? And make these really goofy things. And see, would that translate into this phenotype or in another phenotype? Often it was a huge mess, right? But sometimes it actually, you learn from it, and you couldn't do this in more complicated systems. Eventually, at that time, if you wanted to get a bigger paper, you had it to show all sorts of things. That was also very funny. I had a very, very talented PhD student, Nariman Balenga. He's now in the States, and he's a professor himself. Wow. He did an experiment, really exciting, also in hex cells, and then in human immune cells, right? Because we had blood donors at the department and all that, and we showed the same thing there. It was wonderful. Sent the paper to really high profile paper, and they wanted us to do this in mouse cells. And we were like, wait a minute, okay, we are studying a human receptor. We have this actually in human cells, and you want us to, from human donors, and you want us to prove that this is the same in the mouse, although the mouse receptor is different than the human receptor. That doesn't make any sense, right? It's hilarious. Well, it was sad because we couldn't publish it there. We published it somewhere else, really nice. But that shows you how sometimes even editors of big paper, I think it was Blood, say, they don't see, you have to do it in this more artificial system before we can believe what you see actually in the human cells. That was the other side. It wasn't so much fun, I would say.
Yeah. Oftentimes the comment is, what is the physiological relevance
Yeah, and we showed it, just not in the mouse model. So,
yeah, I can also kind of relate to that when especially when you're we're going to wrap up the Hexcell topic very soon, but what I was going to say is that when you work with Hexcells, usually the big comment that you get is that, oh, you're overexpressing.
Yes.
And the reason you get the phenotype that you get or the responses you get is because you're overexpressing. So we did get these comments almost on a regular basis. So the ideal, the way we were also showing it is taking PBMCs, for example, and then show in PBMCs or also differentiated T cells, and show that in T cells, the level of expression of the receptor is pretty much similar to what I'm transfecting. So there you go. And it's not a mouse model, definitely not. But then again, mouse receptors are very different depending on which GPCRs you're looking at.
Well, there comes the systems biology again, because what we can do once we have a functioning, validated receptor model, we just tweak, we just change the receptor number, and then we can predict how the response would look like if you have way more receptors or way less receptors. We do experiments with inducible systems to verify that this is true, but it's actually it's highly predictable.
Wow, that's fantastic. So we're talking about tools, Hexcells, great tools. We love our Hexcells. It's a good system to at least get an idea on what happens. What are some of other tools and initiatives that could help speed up, the identification of new drugs, targeting?
It's do high throughput crystallography. I think this is happening now. The Swiss fell here in Switzerland, Gebhard Schertlow's group and the people surrounding him, they do an enormous amount of high-speed stuff. So everything high-speed to get a lot of data is good. I think biosensors, of course, are great, but you need biosensors also in primary cells and tissues, etc. I got really one of the big things when I was still working in the lab was when Brigitte Kiefer for the first time showed a GFP mouse. I think it was for the DOR, the delta opioid receptor. And I thought, oh, that is so cool. Now we can follow the receptor in every tissue in this mouse. Or like EPEC sensors were put into animals. So you could actually look at you could have it much easier to see what's going on in real time in there. And of course, Michel Bouvier is working with his biosensor empire on together with Domain Therapeutics on getting all these biosensors into everywhere and making them small enough. And Kevin Pfleger is doing the same. These are these wizards that make our life much easier in the future, I think, to understand what's going on. I think we need more tools to really understand what's going on in the tissues of at least animals to begin with, to find out if these compounds really do the stuff there that they should do, just to make compounds more safe.
Yeah. And on the other hand, we also getting these tools into disease tissues or cells that come from donors who have, I don't know, diabetes or
exactly
any type of diseases can be very helpful to
kind
of make that kind of have that I want to say a table with two columns where you say this is what happens in a normal cell, this is what happens in a disease state when you have a patient with these characteristics.
Exactly.
Allows kind of to have this this more precision medicine type of approach to it. What do you think are some of the key information? I know we talked about this, but if we can get back to this a little bit, the key information that I'm missing that would help, drug discovery or novel drug discovery to help, not necessarily GPCRs, but if we can chat about GPCRs, that could be really something to think about.
It's what I said before, that the key things is to look at really the interaction of these receptors also with not only GPCRs, with other cellular proteins in there. I think you really, this brings you all to like these fancy 3D tissue cultures where you look at also the more, the 3D structure of an organ to make these mini organs and stuff like that. I think there is a lot of things is happening there to really get these assays set up, and so that you can actually jump from the Hexcell to understanding what's really needed in what kind of tissues do interact with each other and how do they interact with each other. I think this is actually a big thing that's happening I know in many labs, but also in big pharma. where they work on these kind of things. So I think this is more or less, you really, I was asked the other day, what receptor would you love to work on? Where do you think we could do big things? I said, I think the low-hanging fruit has been picked everywhere. There's still some orphans out there, but David Loren will de-orphanize them all for us with his computational wizardness. So I think there's really, it's getting harder to really pick one receptor, and that's the holy grail. You have to do more than that and understand what's going on. Really, that's really where I think is a big and huge challenge. And when I was a postdoc, I worked on receptor dimer ligand, which later turned out to actually be just a biased ligand for one of the receptors I was working on. Nevertheless, and I went to give a presentation at a small biotech startup that actually started knocking out every single orphan GPCR and make a mouse. So they knocked out whatever they could, and they made the mice, and then they looked at the mice, what they were doing. And I came there and talked about dimers. And one of these guys was really angry in the audience, and he really was grumpy and really asking nasty questions. And afterwards we went for lunch, and he said, you just made my life so much more complicated, because we are studying one receptor being knocked out, and now you tell me it's actually two together do another thing. And that's, he hadn't, it was in the very early days, and he was just, okay, that's really complicated. So that's what I'm saying. You need to look at what else is there, not only a GPCR. There's, transporters, all these lovely other things happening.
Definitely, and having multiple GPCRs expressed in the same cell.
Yeah.
What also determines the function is where are the ligands coming from, what are the intracellular effectors that the receptor interacts with, and other people a lot of times, for reasons of simplicity, which I understand, focus on G protein versus beta arrestin. That's not necessarily only the case. Understanding also that, for example, G proteins can go into the nucleus and be transcription factors is also important.
Yes. Yeah.
But also the concept of a receptor being internalized and still being able to transmit signal from in there is also something that needs to be taken into account, which I find really fascinating. And
that we tried to model this, and we could omit it in almost every single model we made.
Oh, really?
But not saying that it's not true, but what I'm saying is that probably the interaction of how we measure and analyze things, we measure internalization versus cyclic AMP in real time. I think this is per se already taking this into account. So, but just to tell you something, we often get the question, oh, what about intracellular signaling? It's like, we built a model with both, it didn't matter, right?
Okay.
So that was really funny. But for the GPCRs that we work with, where there is doubt whether they really do this, so just to be not offending anyone.
No, I don't think so. I think it's so interesting. I think this field is so interesting because all these receptors share common characteristics, yet they do so many different things, and they control so many signaling and cellular processes, which I think why all of us are fascinated by the receptors. And yes, I was thinking with the intracellular signaling, I was thinking of the PTH receptors.
Yes. That's so cool. And you have two endogenous ligands that do two different things, and that is fascinating. So that's very cool. Yeah.
And they're quote unquote small peptides.
Yes.
Which is also an extra cool step. Let's get to our segment 3 right now of the podcast. So you had an amazing career going from academia to big pharma and now to biotech. What is your advice to junior scientists who want to contribute to the field, but also kind of understand how to transition from an academic setting to a more industry-like setting?
Yeah, I've been asked it a lot, and I actually also sometimes I'm asked to give little chats and talks about this to people and to students. I think, amazing career back and forth. I always was intrigued by these proteins. So my career was governed and driven by, oh, this is cool, I want to work in this lab, or I want to work on that, or now I want to work for a big pharma, and that's a cool job, right? So I think the passion should guide you. You shouldn't strive to become a professor, or you shouldn't strive to become, rich in industry, or start your company because whatever. I mean, it could be a motivation. Every person has a different motivation, but it's not always been fun and not always been easy. I think I moved my household, I don't know how many times over how many oceans. In my life, and it also has a bit of a toll on your life to do that. But so you need to be passionate about what you do. So my single advice to everyone asking me this is, do what you're passionate about, and the opportunities will show. Because if you're passionate about something, I strongly believe you're good at it. And when you're good at it, then all the doors will open for you. Changing from academia to industry, I mean, I did my first postdoc in Denmark in Ture's lab, and he was at that time an advisor for Novo Nordisk. Half of the people that were at Novo told me about the jobs at Maria. That's your job. It's because they were colleagues of mine when I did my postdoc there. Networking, right? So I think it is really. For me, it was just an exciting opportunity, not because I wanted to go to big pharma. It's because this was a really cool job description, and I thought I could actually do this. So, and also biotech, at the beginning, it's like, oh my God, am I really gonna do this? And I called Fiona Marshall before I actually decided. Well, I called her for advice. Say, Fiona. She also went from GSK and started at Heptares, now Sosei Heptares. And she said, how was did you ever regret it? And she said, actually, the first change I made was not to Heptares, but another company, Biotech at that time. And it was a disaster. But I learned so much by doing it that I was ready for Heptares. And look what she has done, right? Yes. So she said, it's going to be fun. You're going to love it, right? So this is what I tell every young scientist: follow your passion and doors will open.
Thank you for that. I did have a nice chat with Fiona, I think last week.
Yes.
The episode will come out soon. And yes, she's just fantastic. She has great advice as well. And I think she has a phenomenal grasp on the drug discovery process, GPCRs, and she had as well a great career.
I call her my female GPCR rock star.
I think she absolutely deserves that. So you were mentioning just a minute ago that you have to follow your passion, and you shouldn't look at the job title or look at becoming rich CEO or something like that of a company, but you should follow the science and your passion. What would be your advice to GPCR scientists who have a great idea and they want to, launch a GPCR focused company?
Yeah, don't do it. No, actually do it. No, I think that's a really good question. I think you have to be aware when you have a great idea. You're usually a scientist who has a great idea. That is only a very small part of the whole thing. You need to be able to sell the idea. And you need to be able to do anything but the science, okay? You need to be able to really start building up an infrastructure and doing all these things. So my advice would be, don't do it too early, okay? Have a bit of an experience, also very important in team management. As every young PI, I was like, oh my God, I wasn't trained for this. And it's becoming more and more challenging over the years because you sometimes have to be tough and you sometimes have to be focused, especially in biotech, on the success of the company. So you have to actually also envision that you have to do a lot of things that are soft skills and you better have them. Because it often happens, especially for young biotech startups, that the founders at a very 2 or 3 years into the whole thing, they are simply not able to hold up with the experience level that's needed to run the company. So if you do this, if you really have an exciting idea, find yourself very experienced management that can help you to get the funding and the running of the company done. Don't try to do it all on your own. I think this is a really big warning. People can do it, of course. We know people, Steve Jobs, and people who did it, but eventually they were also taken away from the things they love, which is the science. So I love all of it, but I'm very happy we have a very experienced senior team now, and I can actually now go back to the more science-driven part after a couple of years of floating around. So it's really, really important that you don't lose that out of your sight. Just a good idea can go bust because you don't have the infrastructure around you that you really need, and look for someone who can really help you there. That's the really big advice. And just because your professor tells you, I think you shouldn't stay in the lab, you should fund a company, I think that's not good. I think you should really look for people that can help you there.
Yeah, and also look for mentors who have gone through the experience.
Yes, absolutely. Yeah.
Very important, because yes, as you mentioned, the soft skills are also very important.
I think they're as important as being a good scientist is to have good soft skills. And I'm not saying I have the best, but I learn every day. But it's really challenging to jump from a small lab, maybe of a couple of people that you have supervised, to a company and to stakeholder management, and that's not always easy. You can have funny investors, you can have grumpy whatever authorities, and you have to deal with all of them. Okay, and you don't have a big infrastructure like a university behind you or like a big pharma behind you. So I think this is a really big thing. Find a really cool team and go with it. But go with it. It's good fun.
Absolutely. And I think it's also a matter of building those skills and understanding that having a good idea is important, because you have the foundation on what to build the company on, but understanding also that you're going to make mistakes, and getting those mentors to try and avoid those mistakes, and then the experience will also help direct that. But
I think there's a lot of incubator space now everywhere, and there is also funny thing is our new CEO, he actually is from the States, and he said he has a small consortium of colleagues that saw that actually small startups, they don't need a full-time CEO, okay? They just need an experienced person that can help them in these most important points, and you don't need someone to be there 24/7. You just need someone who really has the experience, and then you can more or less self-run it. It's a really cool, rather new concept, but I think it might eventually work out because there's so many people out there that have retired and that still want to help. And want to help people start up and can give great advice and great experience. And I think this is hopefully in the future this will come up much more.
I hope so too. And you're right. Right now there is so many incubator spacing budding around, at least in the US and in Canada as well. And at the same time, so having the infrastructure, good lab infrastructure when it comes to the science, these spots give you, these incubator spaces give you that. But they also help you, get funding and get mentorship. And universities more and more have these infrastructures because it's beneficial.
Yeah, I think ETH Zurich is definitely, the founders of our company, they went through these trainings for 1 or 2 years, but sometimes that's not enough, right? Sometimes you need a little bit more than that. So, and there, more senior people and mentors come in, but there's coaches, of course, but you can coach only that much. So I think, but usually, I've seen many successful startups happening, and they pop up here almost every week, even in Corona times. So, but then, also know when you start a company, it might not work out. I mean, hell, ours mightn't work out, but I always say to everyone I hire nowadays, I say, at least for now we are having fun while we're trying to keep it alive. So, and that's more or less the motivation, I think, of the entire team right now. We are enjoying what we are doing right now, and that should be what you really do, right? Work to have fun, not the other way around.
You can give yourself more to what you're doing if you're enjoying what you're doing, and also enjoying it with a team that you like to be around, that complements itself, and you can still
we have the funniest team, right? Half of us are mathematicians and the others are biologists, and that's really good fun. It's very different kind of thinking, and it's really interesting to discuss sometimes, like, what are you talking about? So really is, but we're closing in on a common nomenclature, I think. Yeah.
That was going to be to your point. What I was going to say is having people from different backgrounds leads
and
to
different countries. Yeah.
Yes, absolutely, and leads to very interesting discussions. I was at some point in a meeting where a computational biologist, made a computational biologist joke, and everyone who understood it was laughing, and us on the biology side, we were like
what
are you talking about? It must be funny if all the bio people and ML people are trying to get funny, but it wasn't. It wasn't for us, because at that point we were very young, so we hadn't developed that common nomenclature. So diversity is important, having people from different backgrounds, from different countries. How do you think we could increase diversity in the field?
I think diversity is already in the field, don't you think? I think it's just if you say female, male, or whatever. I was at Gordon conferences where there was not a single female speaker in the old days.
Wow.
And now they're all. I think it's already there, and also from different backgrounds. We have a lot of people from Baltic countries, and it's Russians. It's really good fun. So I think that the time also, the world also becomes smaller despite Corona. Especially now, I think it becomes closer because we all talk via, Zoom and whatnot, and Teams and things like that. They can work anywhere in the world, so you can actually work with whoever you want to. We have employees employed in 3 different countries.
Wow.
Or stationed in 3 different countries because they more or less just are on the computer, but we talk every day and we know each other really well. So I think to increase diversity is, I have to say, if you go back, for example, into the upper league of companies or whatever, there is still not enough women or not enough, people with ethnical backgrounds. Yes, it's an issue, but I also think it's changing. We just have to be a little bit longer, more patient, and it will happen. But from when I started, it has already changed. The landscape has already changed dramatically.
Oh, and
so I say there's hope.
Oh, definitely. And these people at the top level right now will have to retire at some point.
Yes.
So we can in some way take their place.
No, I think it's getting better also because also childcare in many countries, has changed, and women are more supported in that. And so it's just a generation that is growing up now. I think has a very different. It will have a different speed into these functions. I think. I hope.
I hope so too, and I hope that the whole question of childcare and flexibility will also come onto the US, which I think is far behind a lot of countries. And you don't have to go too far. Just go to Canada, and it's completely different. Europe, it's also a different way of looking at it. So hopefully that will change here as well, and then we'll get
at a point.
Yeah, I think so. And I think on the long run, employers are starting to understand that you're not just a number, you're a person. And employers are also starting to understand that in order for you to be as productive as possible in the time that you're spending at work, you need to have a life, and you need to be able to, relax, because as a scientist, at least, I got the best ideas in the shower or while walking the dog. And not trying to do something in the lab, but also getting that headspace.
Exactly. Yeah. No. And I think actually this is the good thing about the whole Corona crisis is that, I have actually been in home office for several months now, and I know more about the data of my people than when I was there. Because we have a lunch meeting every single day where we just catch up, and I get informed, and then I have separate meetings with people. The labs were mostly shut down as everywhere in the world. So we had a lot of time to chat and discuss. And I think it's been much more efficient, and I have the lake 2 minutes from here, right? So if I feel hot when it's lunchtime, I just go for a swim and come back, and I'm like, wee, who's next, right? So, it won't stay forever like this, but I think it's actually changing also the work environment a little bit. Also going to meetings, I think still meetings are important, that you meet and have a, up for a session drink with people and do that, and we cannot completely forget about these meetings and how important it is, but it is easier to actually acknowledge now that working from home is not such a bad idea.
Exactly. And it's not only it's not a bad idea, it's also feasible.
Yes.
And it can be productive.
Yes.
And still, you mentioned that you have a lunch meeting every day with the team, and it also, I wonder if that also, well, I'm sure that also builds up that extra trust within the team, and also has everybody come together. And if someone's feeling down, then you have 14 people who will tell you, hey.
Then if I notice this, I just like, call right afterwards and say, what's going on. And it's more or less, it's not the same as you see them, but I think I'm also more aware of what's going on. And that's I really like that. But it's good, when you first time come to the lab again and people, I haven't seen you in such a long time. It's different, right? We're social animals. So, sometimes we really need to see each other too.
Oh, yes, definitely. So when are you going back to the lab? How is it going to happen when it happens?
Well, the plan is now Switzerland finally introduced masks in public transport. Before, I have to take 5 different means of transportation to get to the lab. It's unfortunately 1.5 hours away. But now they introduced that, so now I dare to go on public transport again. It was slightly ridiculous for a while. And so the plan is to go, at least once a week to meet with the team. But we also have a bit of a satellite place now in Zurich itself for the management to meet. So we don't have to go 3 hours every day just to meet, right? So this is how it's going to be for a little while now. But at a point we grow. We also need to move because we're running out of space, and we are still incubating in ETH site here in Zurich, and they want to kick us out at a point as well. So all these things are happening, and if any big investor is listening to this, we are starting a new financing round in September. So if you're interested in investing in our fabulous company, call me or email me. So all of these things are happening now as well.
It's super exciting. So with the episode that we're recording currently, there will be a web page, with obviously all your contact information. So if anyone wants to chat with you, work with you, invest, that
anytime.
Place to,
I love to chat. So bring it on.
That's phenomenal. I have one last question for you.
Yes.
Getting back to the science. So I asked this from all of my guests. What are the top 3 or 5, depending on how many you had, aha moments as a scientist that kind of shaped your trajectory, other than being told now you have to work on GPCRs and not on cell cycle?
I think to really, when you're young, you have to build confidence, right? And I had some very, say, dominant supervisors, right? Mischa Freism was a fabulous pharmacologist in Vienna. He was super smart, but also very, and then Tua definitely was challenging in that respect as well, but good fun. I think the first real aha moment was when I submitted a paper to JPC as a corresponding author, and had little help from Tua, was scanning through it and whatever, and then it got accepted. And I was like, wow, I did it, right? It was my feeling of like, I conceptualized the study, I ran it, I wrote the paper, I submitted it, it got accepted in JPC. At that time, it was big, right? That was a big aha moment. It's like, I can do this. That was probably one of the big things in that respect. The other one, I think I mentioned it, is when I managed to go from a hypothesis-driven Hexel experiment to an animal experiment, and it worked. And I predicted what's going to happen, and this is what happened. That was a huge aha moment. It's like that I can actually, make it work so that it actually even works, in a whole organism. That was really, really cool. I think aha moments that probably were the two most, exciting moments in my scientific life, I would say. And of course, we got a huge EU grant last year with the company, with the help of all the founders in there. It's huge amount of money. We had to pitch in Brussels. You had to pitch for exactly 10 minutes, and you had a really tough interview there. And then we walked out of there, and we all looked at each other and said, but, I think we did nothing wrong here. I think we have to get the grant, because we felt it was just so good, and we got it. So that was probably, biggest grant I ever got in my life, with the help of the co-founders, of course. It was amazing. So these are the big things where you're on the right track, right?
Yes.
Stories. Yeah.
Yeah, it validates all the efforts and all the energy that you put in, and it's also it's a great confidence builder, which allows you to go to the next level.
It is, and especially the last one, the big EU grant. We had all different backgrounds. We had the mathematical guy, we had the business guy, we had me as this little more biology geek, and then there had this whole panel of big investors sitting, an EU panel in there, and you're grilled from all sides. And this team effort, I think that was just so cool, that you can come with, you're only as good as the other people that you're with when you go into this. And this is maybe also a big farewell message. Is like, you cannot do it alone. You always do it with people, find exciting people, partners to do it with you, because the more exciting stuff is when you can join together in it and get it together, right? That's why Biotech is your tiny little team. Will we make it? Hopefully, but we make it together, right? If we fail together. But I think this is important. Alone, do it alone. Sit in your lab on your own. I don't believe in that. Science is too big and exciting to not do it with a bigger team.
Absolutely, could not agree more. On that note, thank you so much, Maria, for your time.
You're welcome.
I really enjoyed talking to you, and
thank you for having me. That was good fun. And it's actually a great podcast. I love this. In the beginning, I was like, Dr. GPCR, what is that? But it's such a good idea. I really thank you for doing this, because you bring the world and the people closer to everyone who's out there. Thank you very much for
Thank you. That's the hope of bringing all of us under the same kind of umbrella, where we can all work together, kind of like a big team.
Yes.
And the goal being improving human health and advancing knowledge on GPCRs. And the podcast, I thought it would be a great way to use a new outlet for people to learn about the science. And I'm really happy to have you. I don't know if you knew, this is one question I did not ask you before, but we do have a career page. One of the challenges I find when you work on GPCRs and you want to continue working on GPCRs is finding GPCR-focused positions.
Yeah.
If you do have positions that you want to advertise, we do have a career page, drgpcr.com/career. It's free. Just send us the link and we'll happy to put it on. It's also a community service at this point to have people.
Super. That's a great idea. Yeah.
Thank you. To find their way into the GPCR world for a long period of time. All right. Thank you so much, Maria.
Thank you. Bye-bye.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Subscribe to the podcast wherever you listen to podcasts. I'd like to thank our guest, Attila Forrest, Jin Chong, and Shivani Sajdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. We're always excited to hear from you. Visit us at drgpcr.com or send us a note at hello@drgpcr.com. I also wanted to wish you happy holidays and a very happy new year. I'd like to thank this year's guests and thank you for being such a wonderful audience. Thank you again for the privilege of your time, and until next time, stay safe.